Today we're going to have a lecture about malaria, and malaria is the second of 3 lectures that we have on specific diseases. And the important thing about malaria, well, there's a number of important things about malaria, but malaria most significantly is a protozoan disease. It's not a mosquito disease, it's a protozoan disease, and I think that's really important to remember. So in today's lecture, we're going to talk about the principles of protozoans. We're going to go back and remember what the characteristics of protozoans are, and then we're going to talk about mosquito biology, and then we're going to talk about some interesting phenomena that probably you're not going to know that much about, or maybe you will, but we're going to take a little different slant on malaria, and we're going to think about it in relationship to coevolution of other traits that in some cases may be deleterious, but in other cases may be protective. So let's begin our lecture. So remember the protozoans, they are one-celled organisms, and they can be free-living or they can be parasitic, which means that they're completely dependent on the host. Now, protozoans, in contrast to some of the other pathogens we've learned about, can multiply in humans, and they can live in the digestive tract, for instance, and they can also live in the bloodstream. But remember, one of the common principles that we've reviewed throughout these lectures are, how does a pathogen get to the digestive tract? Well, logically, it can really get there by, contaminated water and or contaminated food. And that's obviously, when you eat contaminated food, where does it go? It goes to the digestive tract. And the same thing with these blood-borne pathogens. How do they get to the blood supply? Exchange of blood products, direct exchange of blood products. They can be sexually transmitted, exchange of bodily fluids, or in the case of malaria, they can be transmitted by an insect, whose purpose in life is basically to feed on blood, right? And so they do a great job of injecting pathogens directly into the bloodstream when they, conduct their own biological survival mechanism of feeding on human blood. So let's talk more about these phenomena. All right, so where is malaria found in the world? Well, not surprisingly, malaria is found in the places in the world where there are a lot of mosquitoes, in the tropics. Okay, so we see here that in sub-Saharan Africa and in India, most particularly, are the places where these mosquitoes are very dense. And because they're very dense and they carry parasites, they don't just carry malaria, they carry viruses, for instance. They carry dengue virus, they carry chikungunya virus. They're great vectors, aren't they? Okay, so let's talk about malaria a little more in relationship to the fact that, again, it's a protozoa. It spends part of its life living in the human host and part of its life living in the mosquito. Now, there are other vectors too that so-called sting. So we have some diseases that are also carried by flies, for instance. But the flies do not carry malaria, the mosquitoes do. And these protozoans, there's 4 different major species of protozoans that cause malaria: Plasmodium vivax, ovale, malariae, and falciparum. Those are the major protozoan species, and they're carried by various Anopheles mosquito species. And again, where are the mosquitoes most dense? They're most dense in sub-Saharan Africa and in Southeast Asia. So what is the life cycle of malaria? Well, the life cycle of malaria, it is one of the most complex life cycles of all of the pathogenic diseases. And we've seen many. We've seen some that have spore stages that live outside, outdoors, right? And then they have stages in the human host. Well, with malaria, we have 2 stages in the human host, and then we have a stage in the mosquito. So basically, we have 3 stages. We haven't quite seen that complexity yet. So let's take a more closer look at what these stages are all about. So the way you read these life cycle figures is you start, of course, with number 1. So number 1, the mosquito takes a bite, right? They take a blood meal, but in doing so, they also inject these sporozoites, okay? And an injection of those sporozoites is what starts the whole ball rolling. Okay, so then these sporozoites migrate to the liver first off, and they live in the liver for a short period of time. They, certainly further develop into this stage called the schizont. And the schizont lives in the liver. And what do we know about the liver? Just like the alveoli and the lungs, the liver is also very highly vascularized. Why is that? Because the liver filters the blood. That's the role of the liver. So the liver basically is an organ that it has a number of canals of blood that basically bathe the liver and allow the liver to remove various toxins, to conduct metabolic processes, and so forth. So where does the parasite go? It goes to the liver, and in that liver stage is where it further develops, and then it gets to this stage 4 where the schizonts rupture out of the liver directly into the blood supply. So the blood supply here, this is the key part, because when an individual is infected with these parasites, they go to the liver, and when they're in the liver, you cannot detect them. But when they come back out into the blood supply, that's the time in which you can diagnose malaria. So this is the diagnostic stage, and these schizonts then develop into trophozoites, and these trophozoites actually burrow in and live in the erythrocytes. The erythrocytes are the red blood cells. Now, what do we know about the erythrocytes? Well, the erythrocytes are basically a membrane that contains hemoglobin and energy, basically, and that blood, that red blood cell does a great job of exchange of gas and passing nutrients throughout the body. So there are just these wonderful factories of nutrients and oxygen. So they're a great place for these immature parasitic stages to live. So they basically live in the erythrocytes, and then at some point the schizonts rupture, and then they go into this gametogenic stage, or the gametocytes. And if a mosquito then takes its blood meal at that time, it will withdraw. It deposits, but it also withdraws. It will withdraw those gametocytes, and then the life cycle of that parasite will continue. So then we go through various stages of the parasite developing in the mosquito, and this final stage is where the sporozoites migrate to the salivary glands of the malaria, where they get ready to inject another infectious dose to potentially another susceptible individual. So this process is very complicated, but it has, of course, been very successful. We have had the existence of malaria for many, many hundreds and thousands of years, and it's a very successful pathogen, to say the least. So now we're going to switch gears a little bit and talk about sickle cell anemia. So you might say, well, gosh, what does sickle cell anemia have to do with malaria? Well, I tell you, it has a lot to do with malaria. And what we know about sickle cell anemia, we know quite a lot, that basically it's a genetic mutation that is expressed in the hemoglobin molecule. It's basically a mutation where one base pair gets substituted. And because that base pair gets substituted, the shape of the hemoglobin molecule is slightly different. It's a quaternary protein. It's a complex protein. Its function, of course, is to carry oxygen and carbon dioxide to and from the lungs and to the tissue. So if, for instance, we have this sickle cell mutation, what ends up happening is the shape of the hemoglobin molecule is slightly different, and so its capacity to carry oxygen is changed, and also it causes this clumping of the hemoglobin such that the erythrocyte, which is normally a concave cell, it's a membrane with no organelles and basically carries hemoglobin. That concave shape, cell shape becomes a sickle, and that sickle shape is what sickle cell anemia is known for. And originally it was thought that sickle cell anemia was entirely deleterious as a trait. And carriers, now people who carry a double mutation don't live very long in life, but individuals who are carriers, they still experience problems because of the sickle cell, but they're able to live. And so these sickle cells, as I explained, are less efficient at carrying oxygen, and carriers are less capable of traveling to high altitudes. For instance, they're also less capable of engaging in extreme strenuous exercise that requires a high degree of respiration. Now, interestingly, at the population level, carriers of sickle cell anemia are protected from malaria because the malaria parasite, that parasitic blood stage, is not successful. Those parasites cannot live in the sickled erythrocytes. They can live in normal ones. They cannot live in sickled erythrocytes, and therefore carriers are protected from malaria. Interesting, isn't it? So what else do we know about malaria? Well, interestingly, malaria has evolved. So the parasite, the Plasmodium protozoa, has actually evolved along with humankind. So there are other species of protozoa that infect other primate species. So the Plasmodium, I can't even pronounce that word, but there's a very characteristic Plasmodium that infects chimps, for instance. There's another Plasmodium species that infects apes. And it was thought that way back in the primordial prehistoric days, where we were all more similar than we are now, that basically the primates at that time were actually all infected by potentially a similar Plasmodium. But then when we split the human lineage off from the chimp and the great ape lineage, then what also split was the Plasmodium species. Now you might say, well then what's going on in India? Well, India also has its own indigenous primate species, the langurs and the macaques, and potentially humankind evolved from them as well. So the human plasmodiums may have evolved from the macaque and the langur lineages as well, although, it's thought that all of this occurred in the way back prehistoric days, where it's thought that humankind actually arose from Africa. So all of this is very interesting because it really helps us get a better understanding about how these pathogens co-evolve with humankind. And getting insight in this way helps us to think about ways in which we can eradicate disease. So what about malaria pathogenicity? So the incubation period, remember I told you that the detectable stage of malaria is after 7 days. The shortest is 7 days after the original bite from the infected mosquito, and it can go all the way up to 30 days. The symptoms caused by the asexual erythrocyte is such that fever stage, body aches, resembles a respiratory infection, resembles a flu. There are several, if you go to the CDC, they have a whole listing of symptoms that are thought to be associated with malaria. It's a pretty challenging illness to diagnose just by symptoms, because these symptoms resemble other diseases as well. And some of the parasitic stages, or some of the plasmodium species, actually can stay in that liver stage. And remember, in the liver stage, they're not really detectable. And this dormant liver stage can reactivate and cause disease months after the initial mosquito bite. And I think I talked in one of the other lectures about that. My father actually spent time in the tropics. He worked for an aerospace company, and he was assigned to Indonesia and Southeast Asia. And he actually got malaria as a young man. And I remember growing up, every now and then, he would come down with this unknown or, indeterminate flu-like illness, and he would just have to stay home for a few days with fever, and then, he would get back going again, and he was fine. So, it's interesting to know how that process occurs. And I also discussed in one of my classes that I was teaching about this, I had a student from Africa who told me about how it's a rather common disease in Africa, to the point that people don't really think that much about it. Now, as I've explained to you, I've spent some time also in India, and it's thought of as being a pretty serious disease in India. So I spoke to a local area physician who's working with us on our research, and he told me, yes, it's considered to be a fairly serious disease in India. So in order to establish a diagnosis, you need to basically demonstrate presence of the parasite in blood, and you can do that by taking a blood smear and looking at it under the microscope. And there are very characteristic morphologic changes that occur to these blood cells that you can actually see visually under the microscope. You can also, do elaborate PCR, of course, but, in terms of symptoms, you can look for whether or not there's a reduction in hemoglobin, evidence of anemia, decrease in blood platelets, elevation of liver markers, and so forth. Again, the CDC has a huge, a place where you can actually look up the actual symptoms and actual parameters that you would use, for instance, to test for the presence of these parasites. So treatment now depends on several factors. It depends on disease severity, parasite species, and where the initial infection occurred. Now, some of these mosquito species are actually indigenous to certain areas. So depending on where you get infected will potentially tell you which mosquito species and which protozoan you might have been infected with, and that may tell you to give one treatment over another, for instance. But also the location and the species, the parasite species, will also let you know whether or not that organism is drug resistant. This is another organism that is resistant to medication, and this is not an antibiotic, by the way. This is a protozoa. So we can have protozoa drug resistance as well. So this is just a partial list of medication. Chloroquine, Malarone, doxycycline, which actually is a chemotherapy drug, and artesunate, which is a pretty new medication. But I do want to talk about Malarone because, as I've explained to you, I've been traveling to India now several times a year. And every time I go, I have to take Malarone prophylactically. So I start taking it a few days before my trip. I take it during my trip, and then I take it after the trip. And basically, I'm taking it through the liver stage into the blood stage because the Malarone prevents the protozoan from actually living in the red blood cells. So I guess I haven't gotten malaria. I hope I haven't gotten malaria, but certainly I have been taking this medication. And the other thing about it, though, is it's very expensive. So people who live in these high endemic areas generally don't have access to this medication. And, we've learned about, the commonality of malaria in these areas. And, some of the other parasites and pathogens that people are infected with can, for instance, reduce the immune system's capability of fighting against malaria. And so it's important. I mean, there's a number of areas of research in malaria that are really important, one of which is to develop a lower cost, effective treatment for malaria if indeed one becomes infected. So now let's talk about some of our questions. What are some of the ways we can eliminate malaria? Well, we've seen that malaria is a very complex disease, right? It has 3 stages. So let's think about the stages. So we have the mosquito stage, and we also have the liver stage in humans, and we have the blood stage in humans. So basically, these are 3 points at which we can attack the disease. We can eliminate the disease, for instance. A lot of disease eradication efforts have focused on the mosquito. So if we can eradicate the mosquito, right, we can eradicate the disease. But eradication of the mosquito is a very difficult thing to do. 10, 20, 30 years ago, we tried to drain the swamps and certainly put in, very toxic pesticides. And that did the trick for a while, but the mosquito is a pretty resilient organism, and they found other ways. And while that's important, right, mosquito eradication is important, in some cases it's pretty challenging to accomplish. So then the other point of attack, so to speak, are, providing mosquito nets. So that's something that the WHO and United Nations has done a pretty good job with in terms of distributing mosquito nets. Now, in the work that I'm doing in India, when you go out to these rural areas, boy, the mosquito density is pretty high. And some individuals do have mosquito nets, but a lot of people out there actually don't. So mosquito nets is one way in which we can certainly protect individuals from being, bitten by mosquitoes. And using them at night, for instance, is a good time because they're very, very dense at night. Another thing that can happen with mosquitoes is you can use these repellents. Now, when I go out to India, I actually use repellents quite heavily, but they wear out, they wear off, they don't last all the time. And unless you're continually putting the mosquito repellent over you, I mean, I've certainly gotten bitten quite extensively by being there. So, the mosquito eradication is a pretty imperfect way to actually, because you only need one, right? You only need infection by that one mosquito. And when you're in these environments, you see how dense how heavy the mosquito density really is. So what about the liver stage and the blood stage? Well, the liver stage is a little challenging to address, although certainly some medications try and get down there to the liver. The liver, what does the liver do? It detoxifies. So it would be somewhat challenging to get a drug there to actually be able to eradicate the parasite at that liver stage, but potentially it could be done. But a lot of, as you saw in the earlier slide about the treatment, a lot of the medications are actually designed or developed to address the blood stage. And if we can prevent the mosquito or the parasite from migrating and living in the erythrocyte, that's certainly one way of being able to treat the disease. If we can eradicate the pathogen from the blood stage, that would be a way as well. But some of the newer publications that I was reading in preparation for the lecture has now thought of, well, if we can treat the mosquito, right, for the pathogen, for the plasmodium, then that might be a way to eradicate the disease as well. So really, every time you think about eradication, you have to think about what is the biology of the disease, what is the behavior of the vector, and these are the places in which we can think about in terms of developing strategies to certainly eradicate the disease. So, what is the second question? What are some of the ways to protect oneself from becoming infected? Well, I just outlined those ways. Number one, mosquito nets. Okay, number two, mosquito repellent. So really, these are strategies, again, designed to focus on the insect. But if we can eradicate the insect, or if we can manage the insect, then we can manage the disease. Now, there's still a lot There's been ongoing efforts to develop a vaccine against malaria. So, and again, where would you develop the vaccine? Because basically what you need to do is you need to develop a way in which the immune system can eradicate or identify and eradicate the malaria disease-causing pathogen, the plasmodium. So what about the plasmodium could then enable the immune system to eradicate malaria? So we need to find a definable antigen. Okay, what part of the plasmodium pathogen can produce a reliable, identifiable antigen to enable the immune system to eradicate malaria? Now, we know, for instance, if you have a misshapen erythrocyte, you will be protected from malaria. But that's a pretty extreme method. You know, obviously science has not found the answer yet, but we're really working hard on it. So that concludes our second disease lecture on malaria. Thank you so much for visiting educator.com.